So, I mean, just heard a series of just amazing techniques and results that are coming from them. I mean, if we were to move forward in this kind of initiative, I mean, almost each of you, which is working in it with different techniques, I mean, it's almost overwhelming. I mean, what could if you had to pick one thing or two or three things that such a resource or an entity could contribute to what you're doing, I'd be interested in hearing what those things would be.
Okay. Well, definitely, we can use a lot of technologies presented or discussed in this symposium. For example, we're not only working on the 3D structure for the molecules, we're also working on the networks. And even for the 3D, the methylation stuff I talked about, we certainly dream to have this virtual cell, then we can basically just visualize all those, how exactly those interactions occur, and for example, particularly those chromatin-related enzymes, we want to see how they work to remodel the chromatin structures. Yeah, also a lot of softwares we developed, we want to put that online to make it publicly available and share with different people.
Yeah. I would like to say that I think that if you are concerned with creating virtual cell and the way how, for example, David Goodsell does it, so that means you would like to reproduce what is known already, then probably you don't need much of these techniques, because it's all either in textbooks or in papers. But if you would like actually to make this 3D virtual cell not as a thing per se, but would like to make it interesting for biologists or for people from medicine, then you actually should connect it to a discovery moment. Not just making a very nice textbook or a very advanced one, but really to show how these things can be used for molecular discovery or for cellular discovery. And I think when you're getting involved into discovery process, then you need these techniques. You need, for example, mass spectrometry. You need other techniques which are like pathway analysis. And in this way, you can really make your 3D virtual cell project very interesting for biologists. And this is the way how you can actually raise a lot of impact and interest in the community outside of this small community which is gathered here.
So one of the things that I'm impressed with is how there is a need for having descriptions of small molecules as part of this whole milieu, that organisms communicate with each other defensively or cooperatively. The cells in the body, we've heard about neurotransmitters. There's all kinds of autocoids and hormones, and most of the drugs we have are designed to either mimic or antagonize those small molecules. So I think it's an interesting challenge, because those molecules are much more dynamic and transient in their existence within cells or between cells.
So. Absolutely.
So I would emphasize that as well. A lot of this technology has just been developed so recently, and just the magnitude of information you can get is astounding. But I'll just follow up on, again, I think we have to create cross-disciplinary environments where, it isn't typical that you have necessarily computational chemists in a chemistry department, or certainly in a biology department, and how we make this easy to do at the academic level, how we make it easy to train students, where you have data analysis people, computational people, modeling people working as teams together, and we train students that way so they think across these boundaries. I think whatever we do, we have to provide a template for the rest of the community, the scientific community, to do that.
So quick question, Andy McCammon from UCSD. I was glad that Don was able to get his pharmacology hat on at the end of his presentation, because I think one of the things that's worth keeping in mind, especially as budgets tighten in Washington and people are looking for value from projects, is the possibility of using virtual cell models as the next step up for structure-based drug discovery, where you might think about ensembles of targets or potential misdirections of small molecules and their activity. Information you might pull out from these models that would be very medically helpful. I wondered if you wanted to expand on that just a little bit.
Well, one thing I didn't have a chance to include in there was pharmacogenomics, and personalized medicine, personalized healthcare is obviously a direction. A lot of the drugs we have, for one reason or another, don't work well in certain patients or are toxic in certain patients where they may be perfectly good. I mean, we all know about the COX inhibitors that were taken off the market because of that, and they're not the only example of that. So I think understanding how the pharmacogenomics, and the only way you're going to understand that is by understanding how a particular receptor or a particular cytochrome P450 works in the body. So I think we need to be able to have models of that and be able to predict which patients are going to do well on a particular drug regime and customize that drug to that person.
Yeah, just to add a little bit on top of that. So people also started to talk about network medicine because before people only use one drug targeted one therapeutic molecule. And now people start to talk about how to model the combinatory effects of several drugs. So on that regard, I think network modeling is really important, particularly for this 3D structure virtual cell. That'll be really, really important to model this. So you have the link from a molecule level, then to the entire cell to the networks level. That will be really important.
So I have a question that relates to data and models. One person's model is another person's data. So the real question is, how much model is in your data? When we talk about crystallography, we think of that as structural data, but it isn't. The data is the diffraction patterns, and the structure is the model. And these are experimental approaches, so I'd just like to get a sense of how much is model and how much is essentially kind of raw data.
Well, I'll give a very simple example. With the small angle scattering, when you take it from that scattering curve to the P of R function, you've gone into a model because you've had to specify one of the parameters, which is what the maximum length you think that molecule is. And sometimes you're guessing wildly, sometimes it's pretty obvious, but you're beginning to model at that point. So anytime you do anything to manipulate the data, I think you're starting on the path of a model.
Right. So also, for example, in our case, we generate our own data, so-called the peptide microarray binding affinity. Then we also model them. But I think Marcus gave a good example today, earlier this morning. He showed us the deviation between the KCAT. They transfer the KCAT from another organism, and if they did the experiment, they found that there is a huge discrepancy. So I think we probably, of course, the experimentalist always trusts the data, but when you do for the theorists, they probably only trust the theory. But there is definitely a crosstalk or marriage between them. Then you probably have to do this in a cyclic fashion. Then you do the modeling, then come back to the data, and then probably you need to filter out more noise from the data, then do a better modeling.
There's recently been a competition that Merck had, which was for a prize for who could predict binding of small molecules to targets. And it was won by a group at University of Toronto. It was actually led by a graduate student of Jeff Hinton. And they had no knowledge whatsoever of the domain. They didn't know anything about the data set. In fact, they were just treated the features that they were given as numbers. And they were able to beat the best teams by quite a large margin using machine learning techniques, and specifically deep networks with many layers. And I think there's a real opportunity here because there's been a real breakthrough in being able to train up these deep networks, even with small amounts of data. And machine learning basically uses pattern recognition, but in a very high dimensional space with very complex and abstract features, the kind that are useful for object recognition and vision and speech recognition, which are also making major advances. And I think this is something to keep in mind. As you collect more data, big data basically gives you the opportunity to use these pattern recognition techniques to make progress. And Wei Wang's, I think, had an example of how you leaving one out, that cross-validation is one way of doing that, but there's a lot of other opportunities, I think.
Yeah, totally agree.
I also would just comment, I think you use your data to drive your models, and we've relied a lot on crystallography for the high-resolution models. But those are static structures, and often they're not complete molecules either. And it's very challenging because these are so dynamic to get a really high-resolution structure of the whole molecule. And so I think you need to be able to model the dynamics as well. For that, you're going to need the low-resolution, more low-resolution data. And I'm also thinking, you know, we have been doing a lot with cryo-EM and reconstructing these large complexes, but I think with the advances in microscopy, that we're beginning to actually see molecules at the level of helices and things in live cells. And I think that that's where you want to see it. That's where you want to see these molecules at the synapse, and you want to see how they change. And you can't see how it changes in that crystal structure necessarily. So you really have to scale those different levels and different kinds of techniques. Make sure what you do see in the crystal structure is what's happening in solution.
Yeah, also I like to add is we probably cannot completely rely on the experimentalists to generate the data because the data always noisy. Then just like Terry pointed out, then we probably need to use those machine learning methods. Then also for some modeling, some people just showed if you even you do PDE, then the parameters, the reads can have a huge range, then that probably won't affect your prediction results that much, only affect probably a couple of %. So given that said, then we probably can use some cross-grade model. You don't have to measure every rate, every parameter in your model, but you can have an estimation, have a range, then let the machine learning method to find the patterns and estimate the parameters, and probably that can have you give you some hints for the further experiments.
And I would like to comment on this question about machine learning because my background is actually machine learning. And it's interesting to see how machine learning is now actually spreading into different fields. And I've seen it in many fields that it works in the way that first one team comes, beats everybody, and with no knowledge, like you said. But then actually they are, with a later progress, people take the next step and they marry the machine learning with very well understanding of models. And this is actually the way to go in many, many fields. And I think that here it's also the same. As long as you are modeling your cell in the ideal world, you don't probably need the machine learning. So you can perfectly deal with your models, whatever the models are. But when you would like to make the next step and go to the reality, then the noise, variation, it becomes biological variation. It becomes so large, then you actually are swamped by this. You don't know what to do. And if you like to model it, then you will be doing it for hundreds of years. And then actually you can use it, machine learning, but not just a replacement, but as a complementary way to deal with this variance and so on in real life data. And this is what, again, I've seen in many fields, and I think this is also the way how it will be developing in this community as well.
One also comment I could make is that we haven't said so much about the huge advances in computing and being able to use the game computing methodology to actually make very fast computing available for understanding biological molecules. And I think that you really have the computational methods now coming into a time frame that matches our experimental ability to measure molecular motions in microsecond, millisecond time range. And that's an extraordinary opportunity. You can now validate your models experimentally, your computational models. Yeah.
Yeah, so I just had a question on the 3D imaging, and I find it really fascinating, and I wanted to get your thoughts and also Don's thoughts on how to really monitor drug action, using these sort of 3D imaging techniques, and what are the challenges, computational challenges, and the experimental challenges. So if you can sort of comment on that.
You probably go ahead.
The challenges are myriad. I'm not in a pharmaceutical company, so but it is perhaps. So I'm not confronted on a daily basis with all the different challenges. But obviously, one of the big challenges is selecting a target. So if you're trying to treat someone with diabetes, what is the best molecular target? Or an arrhythmia, what are one of the best ion channels to target? Or do you target multiple? If you're trying to target cancer or a particular type of cancer, what is the molecule? Sometimes it's obvious, and sometimes it's not obvious at all. And sometimes it's obvious, but then when you knock that protein out as a surrogate to inhibiting that target, you don't get the desired effect. So I think the virtual cell, virtual tissues, virtual organisms are going to be essential to helping us try things in silico before we actually take them into humans, which is where the expensive part is, right?
You also have two of the best model cells. One is the cardiac myocyte, where you have a lot of information and a lot of pharmaceutical reagents and normal molecules that are influencing that. And then Terry at the synapse. Again, those are two highly developed model cell systems that have enormous implications for drug action as well.
So, from my perspective, I think that the 3D or not 3D, all these new technologies, they have more or less the same issues, like experimental issues, how to set up, and also computational issues. But I think they're all quite standard. I think that the biggest challenge, if you would like, for example, to consider the drug development, then is actually the will to develop this drug. And so what I see quite externally out of the field, I see that universities, they are not actually interested in it. So or academia, they are not interested in it. So industry is interested in it. So and academia is actually driven by, I don't know, Nature publications or whatever. And but I see now a very nice trend of combination of academia research and industry research, where academia is very flexible, very fast, can develop new ideas or new targets, for example. And then afterwards, academia has really no power to do all the tests that you need to do to really make the step to the drug. But then industry takes the lead and actually finish it. So I think this is actually one of the big challenges, is really to combine it.
You have to push the button for a long time, a couple of seconds. No, you gotta push it longer.
How about now?
Yeah.
Okay, yeah. So I'm in Nomel from UCSD, and I have a specific question for Dan. Like, you showed some small angle X-ray and neutron scattering studies on pKa, and also introduction on GPCR. So I'm wondering, like, would you apply neutron and X-ray scattering studies to GPCR also? And if you do that, like, what would be the challenge over there for drug design? Yeah.
What was the last part of the question? What, applying neutron scattering to which?
To GPCR.
To GPCRs. I mean, I could say Don can expand on this as well, but we're building this collaboration with Oakridge, which is putting online enormous neutron capabilities now, and actually using pKa, and with Heidi Ham doing the G protein coupling, and then bringing in the GPCRs. And I think they have special advantages too, because the membrane, using membranes, you can have special other things that you can use besides just looking at proteins and solutions. So this is a really great emerging area, I think.
Yeah, so neutrons actually are very useful in studying membrane-bound proteins, because the neutrons are scattered differently by proteins and by lipids, and so you can resolve the protein scattering from the lipid scattering. There's also neutron techniques that that are not as widely used because you have to go, well, neutron techniques are not widely used because there aren't that many facilities in the world. But there are techniques for getting lots of different kinds of dynamics information, from picoseconds up to microseconds. And those techniques have not been well exploited yet, but I think we'll benefit from molecular dynamic simulations and other types of simulations to.
So I'm told that we should end the discussion and go for lunch. And I want to thank everybody for the discussions. I mean, we have enough projects to really do much more beyond the boundaries of whatever we finally decide on. So I hope that we've nucleated a lot of new thinking for everybody. So thank you.